On this screen, you see a prokaryotic plasmid with an origin of replication along the dotted line. The difference between prokaryotes and eukaryotes is that eukaryotic replication has many origins of replication, while prokaryotes, there's just one. Another interesting feature seen in prokaryotes is that replication happens in both directions from the point of origin. The first enzyme you see on this page is labeled number 1, and that's the enzyme gyrase. And this is an enzyme that's part of a larger class of enzymes called topoisomerases. And the function of gyrase in DNA replication is to induce supercoiling ahead of the replication fork. And this is a way to reduce torsional stress that's placed on DNA when the replication fork is opened. The second labeled feature is helicase, which you see right here. And in this case, it's responsible for unzipping the DNA. It separates the helical structure of DNA into 2 single strands. The third feature is primase, and that's labeled number 3. And the function of RNA primase is to insert short RNA primers along the lagging strand, which will be explained in the next page. And this allows DNA polymerase 3 to anchor, and I've put an example of an RNA primer right there. They're just placed along the lagging strand. The last feature that's labeled on this page are the single-stranded binding proteins that are along each single strand. And the primary function of these enzymes these proteins is to keep the fork open, which allows crucial enzymes to access the strands, and they also help prevent the single strands from being degraded by nuclease activity. On this page, we see the rest of the enzymes involved. We see DNA polymerase 3, which it attaches to the primers that I mentioned on the last page, and it synthesizes DNA in a 5' to 3' direction, and that is the only direction that DNA polymerase 3 can synthesize in. So the strand where DNA polymerase 3 is able to synthesize a long, continuous strain that goes into the replication fork, we call the leading strand. The other strand is called the lagging strand, and it's composed of short segments of DNA called Okazaki fragments. And an enzyme that's not pictured here, but has similar activity to this enzyme, is called DNA polymerase 1. And the function of that enzyme is to actually replace the RNA primers that were laid down by primase and replace them with short DNA sequences. And then finally, you have ligase, which actually acts as kind of a molecular glue and binds the Okazaki fragments together by restoring phosphodiester bonds, which are the bonds that attach carbons of sugar molecules together, which hold DNA together.